Up next, 30,000 species under the sea. A few years ago, marine biologists working off the coast of Oregon collected samples of seawater from the very deepest parts of the ocean there at two sites near an active ocean floor volcano. And once the samples were brought to the surface, biologists on the other side of the country in Massachusetts analyzed the samples to find out what kinds of microbes were living at the bottom of the ocean, and what they found was astonishing. An amazing array of microbes, more than 30,000 species, many which have never been seen before. They are new to science, and the results of that analysis were published earlier this month in the journal Science. Joining me now to talk more about what they found is my guest, Julie Huber, is assistant scientist at the Marine Biological Laboratory in Woods Hole, Massachusetts. Welcome to Science Friday.
Thanks, Ira.
Tell us, were you really shocked by what you saw?
We were genuinely shocked with what we saw. This was the deepest sampling to date of any environment in the ocean, of looking at the types of microbes there. And we used DNA sequencing technology and looked at almost 1 million different sequences. And we thought we would actually be able to sample all the diversity there, and we discovered we hadn't. And so we are very surprised with these results.
Mm-hmm. Why should they be so different than everywhere else?
Well, it's not necessarily that they're more different than everywhere else. It's simply that we haven't sampled enough in the ocean to even know what's really there. So undersampling is kind of a fact of life in oceanography, especially in the deep sea. You know, we know more about the surface of Mars than we do the surface of Earth because it's covered in water. So we simply haven't gone out there and sampled enough environments and sampled them deeply enough. Mm-hmm. And so what we did is developed a technology that allowed us to sample thousands and thousands of organisms in one environment and begin to make some strong statistical conclusions about who's actually there.
And you collected these at an active underwater volcano.
Yes.
Sounds fascinating in itself.
Yes. They're from a volcano called Axial Volcano, and it's located on the Juan de Fuca Ridge, which is only about 300 miles off the coast of Oregon. So it's one of the best studied seafloor systems in the world because that's only about a 24-hour ship ride from land. And for about the last decade, scientists from NOAA and the Pacific Marine Environmental Labs have been going out there every year to monitor the system. And it erupted in January of 1998, so it's extremely active, and we've been collecting samples every year and trying to follow the changes in that system.
And how did you actually collect the samples?
So we use something called a remotely operated vehicle, or an ROV. So it's a big flotation device with a bunch of arms and collection devices and cameras and lights, and it's tethered to the ship. And we have a group of pilots and engineers on the ship that control it, and we use monitors to see the seafloor, and we collect our samples that way.
Sort of like the remote-controlled robots that went down to the Titanic.
It's exactly like those, yes.
Yeah. Do you think robots can do that as well as people?
Well, the wonderful thing about being tethered to the ship is you can stay down for days and days and days. You don't have to worry about people. You don't have to worry about batteries or oxygen or anything like that. So you can do a lot of very efficient science on the seafloor that way.
Mm-hmm. You found 37,000 different kinds of bacteria, but you also found these archaea.
Yes. So we looked at two domains of life, both the bacteria and the archaea. These are both microscopic forms of life. And we found that we were able to pretty well sample the archaeal diversity. Mm-hmm. And so a lot of people might be familiar with archaea because they're kind of known as these extreme organisms that live at really high temperatures or really low pH, things like that. And we found that we were actually able to sample that diversity. But with the bacteria, we've only gotten about halfway there, and that's with sampling over 600,000 sequences from this environment. So we still have a ways to go.
And that's really a hostile environment down there, isn't it?
Well, you know, there are a lot of different environments at a hydrothermal event, and the specific samples that we looked at sort of represent a mixture. Mm-hmm. So it's where, you've seen those images of black smokers with the hot fluid gushing out of the seafloor.
Right.
And these samples were actually warm fluids that mix with seawater. So they're actually a bit cooler. They're only about 100 degrees Fahrenheit, and they sort of have all the good things from seawater, like oxygen, and all the good things from hydrothermal fluids like hydrogen and sulfur compounds. So it allows a lot of different organisms to exploit those energy sources.
1-800-989-8255 is our number, but they don't have any sunlight down there.
There's no sunlight. This is a chemosynthetic-driven system. So the reason you see all those beautiful tube worms and clams and things is because of microbes that are fixing carbon dioxide that's coming from the vent fluid, and they're combining that with chemical energy like sulfur and methane to make carbon for these big animals.
Is this part of a larger effort to try to catalog all the life on Earth? And if you want to find that life, you got to go down to the bottom of the ocean, too.
Well, this is definitely part of a much bigger effort through the International Census of Marine Microbes, which is part of the Census of Marine Life. And we are trying to understand, discover, and catalog all of the microbial diversity in the world's oceans. So we're not just looking in the deep sea. We're looking at the sunlit surface ocean. We're looking in marine sediments. We're looking at methane hydrates. We're looking at cold seeps. We're looking at estuaries. So we're trying to use this sampling technology to really begin to understand what microbes are out there, what is their distribution, what are their patterns, and how are they changing.
And it's just a fascinating place, is it not?
Yes, it is. The ocean is a great place to do microbiology, especially the deep sea.
Yeah, and you can expect them to find lots more life since we haven't censused yet, or taken its census, to know what's down there.
I absolutely agree, and the more places we can explore, the more we're going to discover, I have no doubt.
We talked to Craig Venter a few years ago about his efforts to catalog a lot of the stuff in the ocean on the surface. Is that part of the same effort?
Well, the difference between what Craig and other scientists are doing is they're looking at all of the genes in the ocean. So they're just they're going in and they're randomly sequencing the whole genome of all the different microbes in the ocean. We targeted one specific gene. And so in one sense, it's even more remarkable because just by looking at one gene, we still can't catalog the diversity. So you can imagine if you're looking at all the genes from all the microbes, how challenging that effort is going to be. We
were up in Alaska and we were talking with their efforts to talk about the Arctic Circle for the first time. They were looking at parts of the ocean that had never been looked at.
That's right.
Is this the same area in Oregon that was never looked at before?
Well, we've like I said, we've been going to axial from quite some time. But certainly if we discover hydrothermal systems in the Arctic Ocean or down near the Antarctic, I mean, we have a lot of mid-ocean ridge that's unexplored. We've only explored about 20% of it. So any new site we're certainly going to be interested in examining.